Hello to all. Hope so you all are well. Today we are going to start with a very important topic of the plant anatomy, and that is known as the ground tissue system. This ground tissue system is basically the fundamental tissue system. And it is called as the fundamental tissue system, and it forms the bulk of the plant body, and it forms bulk of plant body. It forms bulk of plant body means majority part of the plant body is made up of this ground tissue system. Now, in dicot stems, remember this thing, in dicot stems, not in monocot. In dicot stem, dicot roots and monocot roots. Again I am repeating, in dicot stems, dicot roots and monocot roots, there is a clear zonation of ground tissue system. That is, it is divided into two zones. Number one is outer cortex and number two is central pith. Are you getting? In dicot stems, the dicot roots and the monocot roots, there is a clear zonation of the ground tissue system, and it is divided into two zones, outer cortex and the central pith. Fine. Now I am putting a note here. No such clear differentiation is found in monocot stem. Very, very important, monocot stem. Why? As in monocot stem, endodermis, pericycle, pith and pith rays absent. In monocot stem, endodermis, pericycle, pith and pith rays are absent. So we cannot say that the ground tissue system is divided into two zones, outer cortex and central pith. One more thing. In case of leaves, whether it is monocot or dicot, doesn't matter, all leaves, right? In case of leaves, all leaves, I am talking about all leaves, monocot as well as dicot, in all leaves, ground tissue is called mesophyll, tissue, and it is not differentiated into cortex and pith. You will be not able to see the differentiation between the cortex and the pith in the case of the leaves. Is it clear or not? So this is the basic of the ground tissue system you must know. Now, in ground tissue system, we study number one, cortex. In ground tissue system we study, number one is cortex. In cortex, what we study? Hypodermis, then general cortex, then endodermis, right? In cortex we study hypodermis, general cortex and endodermis, and here in the next central pith region, what we study, that is in the steel region particularly, we study pericycle. What we have to study? Cortex. Cortex is hypodermis, general cortex, and endodermis is equal to cortex. Also we have to study the pericycle, pith rays. We have to study pericycle, pith and pith rays. Alright. Now let's talk about the first one, that is the outer cortex. Means we'll be studying first the outer cortex region, that is hypodermis, general cortex and endodermis. So the number one is the outer cortex. We are studying the zonation of ground tissue system. We are studying the zonation of ground tissue system, and we are studying the first zonation, and that is cortex. Now what is the first part of the cortex? First part of the cortex is hypodermis. First part is the hypodermis. Now as the name is indicating, it is present below epidermis. It is present below epidermis in dicot and monocot stems. Remember this thing, this is very important. What I have said you, hypodermis is present below epidermis in dicot and monocot stem. What is hypodermis? Hypo means below, epi means above. Say for, if this is the epidermis, epi means above. This is the epidermis. Then just below the epidermis is the hypodermis. This is the hypodermis. This is the hypodermis. Now always remember that I am putting a note here, very important. In dicot stem, hypodermis made of very important collenchyma. In dicot stem, hypodermis is made of collenchyma. Remember, this all things will be playing a very important role when we will be studying the internal structure of the dicot stem, monocot stem, dicot root, monocot root, and dicot leaf and monocot leaf. All these things will be important there. So just kindly concentrate here. In dicot stem, hypodermis is made of collenchyma. In monocot stem, hypodermis is made of sclerenchyma. It is made of sclerenchyma. And one more thing, that generally intercellular spaces are absent in hypodermis, hypodermis is outermost layer of cortex, because just now I have said you that cortex is equal to what? Cortex is equal to hypodermis plus general cortex plus endodermis. Are you getting? So it is the outermost layer of the cortex. Now, one more thing that very important, in all roots, no matter whether they are dicot or monocot, in all roots hypodermis is absent. Hypodermis is absent. So you will not find the hypodermis in the case of the roots. Alright, in all roots hypodermis is totally absent. And what is the prime function? What is the prime function of hypodermis? What is the prime function of the hypodermis? So hypodermis gives mechanical support. Hypodermis gives mechanical support. As you know very well that whether it is collenchyma or whether it is sclerenchyma, both are mechanical tissues from which the hypodermis is made. The collenchyma is a living mechanical tissue and sclerenchyma is a dead mechanical tissue. So definitely they will be providing the mechanical support. And number two, sometimes when chloroplast is present in hypodermis, it also helps in photosynthesis. Fine. So just now we have started the zonation of the ground tissue system. You know very well that in dicot stem, again I am repeating, in dicot stem, in dicot root and in monocot root, the tissue system is actually divided into two zones, right? One is the outer zone known as the cortex, and one is the central zone, which is the pith. So in the outer cortex region we have studied the hypodermis. Now coming to the next one, and that is known as the general cortex. Coming to the general cortex. Now first of all you must know that where is general cortex present? Where is general cortex present? Alright. So let's talk about the general cortex. Now in dicot stem it is present. In dicot stem, remember, very important, in dicot stem it is present next to hypodermis and extended up to endodermis. Means we can say that in dicot stem it is present between hypodermis and endodermis. Say for this is the tear section of dicot stem. So this is the endodermis. This is the endodermis. And this is the hypodermis. So where is general cortex present? General cortex is present here. This is general cortex. Cortex. So I can say that it is present in between the hypodermis and the endodermis. So in dicot stem it is present next to the hypodermis and extended up to endodermis. We can say that it is present in between the hypodermis and the endodermis. Clear? So it is the section of the dicot stem. Now let's talk about that where is the general cortex found in monocot stem. Now in monocot stem, general cortex in monocot stem, in the case of monocot stem, general cortex extends from below of hypodermis, up to center. In monocot stem, general cortex extends from below of hypodermis up to center. Why? Now the question is why? As in monocot stem, endodermis, pericycle, pith and pith rays are absent. So nobody will stop it. Here what happens in the dicot stem, that general cortex is up till the endodermis only, because here there is a demarcating layer known as the endodermis. But what will happen in the case of the monocot stem? In monocot stem, what will happen? That this general cortex will extend up to the center. Say for these are the vascular bundles. Alright. And this cortex will be extended up to the center. This is the vascular bundle. And this is the general cortex which is extended up till the center. Why? Because there is no endodermis. There is no pericycle, there is no pith, there is no pith rays. So the general cortex, right from the below portion of the hypodermis, it extends up to the center. So remember that where is general cortex found in the dicot stem and where it is found in the monocot stem. Are you getting? Now let's talk about where is the general cortex found in the dicot and the monocot root. So just see here. Now in the case of the dicot and the monocot root, you know very well that hypodermis is absent. So definitely it will be arising from, or it will be found next to the epidermis. So in dicot, particularly in the case of dicot and monocot root, in dicot and monocot root, it is present next to epidermis. In roots, epidermis is also called as epiblema or rhizodermis. In dicot and monocot root, it is present next to epidermis. In dicot and monocot stem, it was found next to hypodermis. In dicot and monocot roots, it is present next to epidermis up to endodermis. Now, as here hypodermis is absent. Hypodermis is absent. So say for this is a root section. So in root, say for this is the epiblema, which is the epidermis, and right from next to the epiblema, because hypodermis is absent, here is the endodermis. This is the endodermis. So right from epiblema up to endodermis, here is the general cortex. All right. Now remember this thing, that general cortex is made of parenchyma cells. It is made of parenchyma cells, and parenchyma cells are living, and it has many intercellular spaces. You can see here many intercellular spaces are there. Now what is the function, what is the prime function of this general cortex? What is the prime function of the general cortex? That the prime function is that it stores food. It stores food, as it has many intercellular spaces. It stores food, and you know very well parenchyma is having the prime role to store the food. When chloroplast present, it also helps in photosynthesis. All right. So we are talking about the zonation of the ground tissue system, and we have started the outer cortex. And in this outer cortex, we have studied the hypodermis and the general cortex, which is actually general cortex is the middle one. Now coming to the innermost zone of the cortex, which is very, very important one, and that is known as the endodermis. So coming to endodermis. Coming to endodermis. Endodermis, innermost layer of cortex. Innermost layer of cortex. Now, one thing to be noted here is that in monocot stem, as I have said you, in monocot stem, endodermis absent. In monocot stems, particularly in monocot stem, there is no endodermis found. Point number one. Number two, now endodermis just below endodermis, pericycle is found. Pericycle found. So we can say that endodermis is found between general cortex and pericycle. Are you getting? This is the endodermis. All right. This is the endodermis, and this is the pericycle. This is the pericycle, this is endodermis, and here what is found? General cortex. This is general cortex. here it is epidermis, this is epidermis, and here is the hypodermis. Fine. This is hypodermis. Alright. So can I say that it is found, endodermis is found between general cortex and the pericycle? Got it? Now, endodermis is single laid. Endodermis is single laid. It is made of living parenchyma cells. It is made of living parenchyma cells. Cells are compactly packed. Cells of endodermis are compactly packed. Intercellular spaces absent. Cells are compactly packed, intercellular space is absent, and the key feature which was asked in the examination, and that was that cells, very very important, cells of endodermis are, question was asked, barrel shaped. Barrel shaped means they are just like drum-like or cylindrical. Just they are like a dholak, if you have seen it, like this type. Alright. So cells of endodermis are barrel shaped. Now, on endodermis, starch grains are also found. Starch grains also found. Hence, it is also called starch sheet. So if somebody asks you that what is starch sheet, so starch sheet is nothing, it is the endodermis only. And why it is called a starch sheet? Because it consists of the starch grains. Okay. Now let's talk about something more about the endodermis. Okay, some special features about the endodermis. Now, the endodermis also consists of certain strips. Endodermis also consists of certain strips. Endodermis consists of certain bends or strips called, very important, Caspin strips. Caspin strips, which are distinct in roots but not so much clear in stems. So now, as I have said you that they are not clear in stems but they are clear in the case of the roots. Now the question arises that what are Caspin strips? So Caspin strips were discovered by Caspari. They were discovered by Caspari, and hence they are called as Caspin strips. Now these Caspin strips are made of suberin, though lignin is also found. but chiefly they are made up of suberin. And you know very well, suberin is impermeable to water, doesn't allows the water. Now, very important, at few places in endodermis, Caspin strips are absent. In few places in endodermis, Caspin strips are absent. Remember that these places are found in front of protoxylem. These places are found in front of protoxylem. I will just draw you a diagram so that the concept becomes clear. At few places in endodermis, Caspin strips are absent. These places are found in front of protoxylem. At these places where Caspin strips are absent, at these places passage cells are found. And as the name is indicating, passage cells are meant for entry of water. Why? Because on the entire endodermis except the passage cells, Caspin strips are found, and Caspin strips are impermeable to water. So now the question arises that how the water will be entering into the root xylem. So the water will be entering through the passage cells. Say for, if this is the endodermis, alright, this is the endodermis, and say for, if I talk about the roots, then this is the protoxylem. You know, protoxylem is found at the periphery. In the case of the roots. Exact condition is that. So what I want to say, that those places of the endodermis, those cells of the endodermis which are in front of the protoxylem, at these places passage cells are found. PC means passage cells. But at all other places you can see, at all other places of the endodermis, what is found, beta? Caspin strips is found. These are the Caspin strips, chiefly composed of suberin. So water will enter through the passage cells, and then the root xylem will be taking this water. Alright. So this is all about the endodermis. Alright. Like this, we have completed the first part of the ground tissue system, which is the outer part, that is the cortex. And the cortex was composed of hypodermis, general cortex and endodermis. Right. Now coming to the central part, central region of the ground tissue system. Now central region consists of the pericycle, right, the pith and the pith rays. Starting with the central region. Now you know very well that central region consists of what? Number one is we have to study pericycle, then we have to study pith, and then we have to study pith rays. All these three are included in the ground tissue system. Now let's start with pericycle. Now pericycle is boundary layer of steel. It's boundary layer of steel. Remember this thing: pericycle, pith rays, and vascular bundles. All are included in steel. Though these vascular bundles we will not study in the ground tissue system, because they are the part of the vascular tissue system, which we will be studying separately. You know now that tissue system are of three types: epidermal tissue system, ground tissue system, and vascular tissue system. So though they are actually the part of the steel, we will be studying separately in the vascular tissue system. So say for this is the pericycle. This is the pericycle. Alright, and all portion inside the pericycle, this one will be called as, including the pericycle, will be called as steel. So say for this is the pith. From pith arises pith rays. So these are the pith rays, and these are the vascular bundles. Alright, these are the vascular bundles. So what I want to say, pericycle and this pith, and then pith rays, and then vascular bundles, all comprise the steel. This is a steel. I will make a video separately on the steel, because the steels are also of different types. Alright, when the proper time will come, I will upload a video on steel also. So we have to study first the pericycle. Now pericycle is the boundary layer of steel. The pericycle, now the key thing to be noted here is that pericycle is absent in monocot stem. As you know very well that in monocot stem, endodermis, pericycle, pith and pith rays are absent. Okay, you have to remember this thing. Now pericycle in dicot stem is multi-layered, made of parenchyma, and sclerenchyma. Again I am repeating, in dicot stem, particularly in the dicot stem, it is multi-layered and it is made up of parenchyma and sclerenchyma. Now pericycle in roots means all roots, monocot as well as dicot. Pericycle in roots is single layered and is made of parenchyma. So in roots it is made up of parenchyma. Now coming to the functions of pericycle, that what is the prime function of the pericycle. So remember this thing, that in the case of where in dicot roots, pericycle, very important, pericycle forms cambium ring. In dicot roots, pericycle forms cambium ring, which forms secondary tissues. And you know very well, secondary tissues are formed at the time of the secondary growth. Number 2, very important question was asked in NEET, that it forms lateral roots. It forms lateral roots. And the third point is that when the pericycle is composed of parenchyma, it stores food. When composed of when composed of parenchyma, it stores food. And remember, this pericycle is known as boundary layer of steel, right? It is the boundary layer of steel. Are you getting? So the first part of the central region we have discussed, and that is the pericycle. Now coming to the next important part, is the pith. Now, pith. Now the central part is called as the pith. Now in monocot stem, pith absent. Remember this thing. In the case of the monocot stem, pith is absent. Point number 2. Pith is made of parenchymatous cells. Pith is made of parenchymatous cells and has many intercellular species. It is having the prime role of storage. It is meant for storage. Fine. Now, in dicot stem, very important. In dicot stem, pith is very well developed. In dicot stem, it is very well developed. In monocot stem, it is absent. Now, in dicot root, comparatively, or I can say it as that in dicot root, pith is less developed. And in monocot root, pith is very well developed. Are you getting? Remember these 4 points. First point is in dicot stem, pith is very well developed. In monocot stem, pith is absent. In dicot root, pith is less developed. In monocot root, pith is very well developed. And pith is primarily concerned with the storage. Alright. And at the last, we are coming to the pith rays. Now, pith rays, where they are found? Found in between vascular bundles. Arise from pith. Like this. Say for this is the pith, and these are the vascular bundles. Say for these are the vascular bundles. Right? Phloem, xylem. Fine. These are the vascular bundles. So in between the vascular bundles, what are found? Pith rays are found. Fine. So found in between the vascular bundles and arise from the pith. Very important. Pith rays are absent in monocot stem. I've already said you that pith rays are absent in monocot stem. Now very important, pith rays are absent in roots, all roots, monocot as well as dicot, in all roots. Instead, they have conjunctive tissue. They have conjunctive tissue. In place of the pith rays, they have conjunctive tissue, and they are found in dicot stems. So they are found in dicot stem, they are absent in monocot stem, they are absent in roots. In roots, instead of the pith rays, conjunctive tissue is found between the vascular bundles. Remember this thing, pith is also called as medulla, and therefore the pith rays will also be called it as medullary rays, and they are meant for lateral conduction of food. They are meant for lateral conduction of food. They are meant for lateral conduction of food. Are you getting? Lateral means like this type. They are meant for lateral conduction of food. So this was all about the pith rays. So students, today we have discussed the entire ground tissue system in detail. Alright, so that you can perform the questions easily. Listen students, if your theory is very much clear in biology, then you can just attempt any type of questions from any book. If you go through all the videos which I have uploaded, then you can easily attempt 95% questions of any book. So try to focus on these videos, because our videos are totally based on the quality and the content. Alright, so today we have discussed the ground tissue system, in which we have discussed the very first one, hypodermis, general cortex, endodermis, pericycle, pith and pith rays. Alright, so thanks a lot. We will be coming soon with few more videos. So thanks a lot.